Okay, so up to question 12 we already done. Question number 13. What is DNA fingerprinting? Mention its application. Very short question but very long answer is required. What is DNA fingerprinting? What is DNA fingerprinting is actually a comparison of the DNA of a culprit and the sample, blood sample or the semen sample or the sputum sample or hair follicle or any other which is found at the crime scene. So it's a comparison between them. So by comparing them we come to know who is the criminal and we can easily detect. As you know, the name fingerprinting has come into being because earlier there was a practice to compare, to find out that who is the culprit by checking the fingerprint. Everyone's fingerprint is special and no two individual in the world is having the similar type of fingerprint. So whenever anybody used to reach to the crime site and used to touch the lock or the latch or the glass or the plates or the fridge handles or door handles or TV remote or anything, any smooth surface, when they used to touch, they used to leave a finger mark over there. Okay, and these finger marks are actually because of some of the cells are always loose cells which keep on shedding from our body, and those cells will accumulate there, they will deposit there, and then using some powder or something, they used to get the mark of the fingerprint rings. But there was a limitation in this. Here you can have only certain limitation, certain application, that we can only find out that who is the person who is holding that gun or who entered the room and all. DNA fingerprinting has a larger scope, and DNA fingerprinting gives the accurate result. Now the normal fingerprinting, the person doesn't make his fingerprinting prepared. He may just touch some hot object, hot iron, and put his thumb, so that this fingerprint mark will be flattened out there, or he may cut it, or he may use gloves or some other way he can stop this type of fingerprint to be left over there. But you cannot fool the DNA fingerprinting, because DNA fingerprinting is a very accurate. Even one cell is there, even a few cells are there, you can compare the cells DNA of from the crime site with the culprit. That is who all are there. That is the people on which we have got doubts that what type of people can involve in such type of act. Their blood sample or their cell samples are taken, and these cell samples DNA and crime site cell samples DNA are matched, and if it is matched, it is the exact 100% sure that this was the person. Okay, now what we have to do, like suppose these are some cells which are left there, maybe from the hair follicle, maybe from the sputum, maybe from because there was a struggle between the person, two people, and some of the cells left in their nails, or some of the cells from the semen, or some cells from the blood, or when some scratch mark is there, some blood is left there, or whatever, some cells are there. These cells are taken to the laboratory and they are treated with a protease enzyme. So protease will dissolve all the protein, the cell membrane, nuclear membrane, the chromosome has got its histone and non-histone protein and all proteins will be removed, and then they are even treated with RNAs. Then we have got only DNA. All 46 DNA are there. Okay, if it is a human, we are looking for the human being only, not for the dog and cat and all, because it's an expensive process. You cannot simply use it on anything. So now 46 chromosomes you got. Out of the 46 chromosomes, certain chromosomes you decide which chromosome you want to take. Suppose you are taking the chromosome number 8. Okay, pair chromosome number 8. Then you are taking the chromosome number 12. Then you are taking chromosome number 18. Minimum 2-3 years to take. Okay. Now in these chromosomes, by the human genome study, we know that where the genes are located. We are not picking the segments which are a part of some genes because they may be exactly same. They may be same in the two people. But between the genes, there are some repeated units. Suppose this is the DNA. This is the DNA. And in this DNA, here to here is the gene, here to here is the gene, here to here is the gene, here to like there are a number of genes. And between these genes, they are non-coding segments. Non-coding segments. These segments are not a part of any gene. They are also known as variable number tandem repeat. Variable number tandem repeat. Or they are also called as the polymorphic segments. Polymorphic segments. Now what are polymorphic or what are VNTR or what are these non-coding segments? Now if I put it this way, initially when a particular species must have come into existence, it may be possible that their genes were one after other. There was no gap. But in due course of time, when the copying is taking place, we have already studied about the mutation. We have studied when the copying is taking place, sometime errors comes. Now if the errors are in the gene, those genes will be defective. They may survive or may not survive. Most often they do not survive because of the errors may be dangerous. But if the errors are acceptable, no problem, the error are not causing any major difference like the curliness of the hair or the color of the hair or the color of the skin or the color of eye or the size of nail. These are generally defects, mutation, and these are not causing any major problem. So they will continue to survive. But there are some major defects may be there, like say for example in place of a diester enzyme protease, some other enzymes start forming, or it may not be a protease at all. So such a person will not be able to digest protein at all. Or suppose the bile pigments or bile salts are not formed, their genes are defective. Or suppose the respiratory glycolysis, we have studied so many enzymes are there. One of the major enzyme is not there. Then the process of glycolysis will not be taking place. How the energy will be obtained, and such person will not survive, or it will be very difficult for him to survive. So in such case, those genes which are there, they will not generally develop any major defect. So we are not worried about them. We are worried about only those segments which are found between the two consecutive genes and which are not the part of a gene, and they are formed because of the repetitive units or because of the number of time copying or cloning of a certain segment of nitrogen bases. So they may have a number of times AAA, then CCC, AATP, like that. So they will be. Now these segments which are there, they are special for that particular human, that particular individual. Say for this is the individual whose name is Rakesh, and Rakesh's father's name was Mukesh, and his mother's name is Priya. Now the Rakesh will get one of the segment from his father Mukesh, another segment from Priya. But it is not always that way also, because sometimes the crossover takes place. So suppose this particular segment from here to here of Mukesh and here to here it is of Priya. So there are some possibility of intermixing, and that is why we have to take the segment from number of places. Okay, but if there like DNA fingerprinting has number of applications, if it is nothing to do with the parentage or the identity for a what do you call descendants, then it hardly matters. Now whatever these segments are there, these are special in the Rakesh. They may be found only in Mukesh and Priya or on their grandparents. Otherwise, no other individual will have the similarity with it. Rakesh, his siblings, brothers and sisters, his father, mother, his grandparents, they may have possibility of these segments. But if you take it from the different places, suppose you are taking from here one and you are taking from here one, there will be very much a possibility of avoiding that chance here. Because Mukesh will have that chromosome from here to here, one single chromosome, but here what he had done is the crossover took place. Now the other siblings which are there, they may also have this crossover taking place in other places. So if we are taking the segments, the VNTR from the different places, even in the same chromosome and in the different chromosome, even the siblings' chances will also be avoided. Their similarity will also be avoided. Okay, so this is what the base of the DNA fingerprinting is. Now, how it is done, let us see. And then we will see the practical aspect. That is the uses. Okay, now the chromosome is removed and that wherever we wanted to take the segment, the polymorphic segment, that will be cut using restriction engine. Using restriction engine, we will cut these segments. We will cut these segments. You can do annealing first and then cut the segment, or you can cut the segment first and then anneal. Suppose this. So now we got these small pieces of the chromosome here and these small pieces of the chromosome here. And this can be separated from the parental piece, that is the larger piece, by electrophoresis. Now these segments will be taken for annealing, that is their strands will be separated, and choose any one strand. If the number of DNA segments are less, what you can do is you can take it, increase the number by cloning. We'll study about cloning in chapter 11. So how the DNA can be cloned and their number can be multiplied. Now you have to put them in the medium containing radioactive nucleotides. So they will hybridize with them, and the whole sequence of the complementary strand will be formed. The complementary strand which is formed is having radioactive nucleotides. So when it is passed through the autoradiographic machine, you get the sequence of nitrogen base here. And by comparing the sequence of the nitrogen bases from the crime site with the culprit, if it is exactly same, we come to know that he is the culprit, he or she is the culprit. So there is no chance of any error, unlike the other probing method may have some errors. Okay, here there will be no error. Now what are the applications? You can use it for simply solving the cases like murder case or the theft case, or it can also be used for claims in the insurance. As I said, sometimes the body burns in such badly that you cannot come to know that whose body it is. People very often do this. What they do is they take the insurance first and then they burn that person's body so badly that you cannot reveal that who this person is, where the actual person may be moved from here to somewhere else, or he may left the country also. So this person's body, how will you come to know that whether it was really that person or someone else? So some of the cells will be definitely there. Those cells are taken and their DNA is matched. Okay, with their parents' DNA or the siblings' DNA. If there is no similarity, that means he or she is not belonging to this family only. Because the original person is moved out. If the original person is there, then how can you say that he is murdered? So when he is moved out, he is not there in the vicinity. In that case, you have to match with their siblings or the parents' blood group or the DNA segment. Somewhere should be similarity there. If it is not there, then one can easily say that this person is nowhere related to this family, so that the claim will be not given. Sometimes the child is exchanged. Sometimes the children are left home or they are misplaced. After so many years they come back, and then you have to find out whether this person is saying truth or he simply want to join the family. So in all these cases, even the last relatives case, that is if someone want to take the visa on the basis of that is the last relative left in country. So how all these cases are actually probed by DNA fingerprinting. Very often it is done by the insurance company nowadays. because people are just taking a claim. Any dead body they say is so and so and it is my son or is my brother or is my this thing, and they'll say कि इसका तो इतने लाख का claim था, give the claim. So the insurance company is not simply going to give you a claim just basis of that dead body of 5 feet or 6 feet and उसी के size का मिल गया and some घर का कोई वैसा वैसा कपड़ा ला कर रख दो, बोले सब ये तो हमारी शर्ट है, मेरी बेटे की है. So these things are not entertained by them. They will simply have a probe. So the probing is done, and the probing is done by DNA fingerprint. So DNA fingerprinting has number of applications. That is for the murder case, for the theft case, for the claim case, insurance claim case, or for the parentage case, or for the last relative left in the country's case, or some other cases. Okay. So these are the different applications of the DNA fingerprinting. Now last is briefly describe following. Transcription. What is transcription? The script has to be changed. How the script is changed? The information is coded in our body in DNA. What I can, I am, and what I can do. All these informations are there on my 46 chromosome, and these information will be materialized. They show the morphology. They will show the features in my body. Okay. Now how it is done? The informations are in the DNA, and the body show physical changes because of some type of enzyme. So the information from the DNA is first converted into RNA, and then RNA is translated into enzyme. So here this particular segment, that is the DNA to RNA, is called transcription. So you have to write only this much. That is the formation of RNA from a segment of DNA is called transcription. Now how is it done? That is, a DNA is very long, and DNA has got 2 strands. Out of the 2 strands, only 1 strand is chosen, and the boundaries are decided by promoter and terminator. The promoter comes and attached to the 3 end of the template strand. 3 ends of the template strand. Suppose this is the everything. So here 3 end, and this is the 5 end. So this is the template strand. So here the sigma factor will come and attach, and then the RNA, DNA dependent RNA polymerase comes here, and this will go on moving from one end to another, and the enzyme heritage will uncoil the segment, and the newly formed DNA will just keep suspended as the process is proceeded. So first thing, that is when the sigma factor come and attach on the promoter, it is called initiation, and then its copying takes place from one end to other, because the new synthesis is always taking place from 5 to 3 direction. So this will be done on the 3 to 5 template, and this is called elongation. And when it reaches here onto the terminator, the sigma factor will change to rho factor. Another unit comes, that is called rho factor, and the newly synthesized what you call RNA will be dropped from there. Okay, so this is how the thing of DNA transcription takes place. Next is polymorphism. Just now I gave a whole lot of information on the polymorphism. Polymorphism is nothing but the repeated unit of the certain nitrogenous bases which are formed during the course of mutation. Okay? And these are not a part of any gene. They are found between the 2 consecutive genes. They are those segments which are not coded. Okay, and the polymorphism in one person is specific. It can be sometime found in the siblings in a certain segment, and they are taken from the parents. Okay, so what polymorphic segments I got in one of the DNA and one of the part, maybe either from my mother or my father, and that segment can even found in one of my siblings. But if I take the polymorphic segment from the different chromosomes in the different segment, then some part may come from father, some part may come from mother, and it may be there. Some segment may only there in the sub-segment sibling, other segment of the same chromosome may not there, because a particular piece of the DNA is come from father, other is come from mother. If you take the polymorphic segment from here, it is similar to the father, it is from mother. My siblings may have this segment, may not be coming from the crossing over. Okay, the crossover must have not taken place here, the crossover must have taken place here or here. So there may be different possibility, or this crossover must have taken place in the polymorphic segment itself. So this is how it avoids the chances of having the same polymorphic segment even in the siblings. Next is translation. This is translation, that is RNA to enzyme is called translation. Now RNA to enzyme, how it will take place? This is the messenger RNA, and the messenger RNA, a small segment of the ribosome will come and attach. It will move from the 3 to 5 direction, and it will look for first AUG. These segments which are not copied, and these segments after the terminator what copies segments are there, they are called untranscribed regions, that is UTR. So first that is codon, it will see the AUG, and when it sees the AUG, this will have two sides, A and B side. A side, the first nucleotide tRNA with the amino acid will come, and then the P side will also the tRNA with the amino acid will come there, and the bond will take place between them, and as soon as the bond is taken place, it will go. So the coming of the location of small and large segment of the mRNA, attachment of this is called initiation. Then again this process will continue from year to year, that is called elongation. Here it was promoter, here it is called initiation. Here it is again elongation here also, and here the terminator. In the terminator, what one of these three nucleotides will be there, either UAA or UAG or UGA. One of these are there, so there is no restricted tRNA for them, and the process will be suspended there. So this is how the translation takes place. Okay, now is the bioinformatics. We have not done a lot about the bioinformatics. This is also a new branch of the biology, comparatively new branch. Bioinformatics what? The bioinformatics is a branch of biology which deals with the use of the computer application for various aspects of the biological processes. Now bioinformatics was first time required when HGP, that is Human Genome Project, was studied. As in the previous class, that is in the yesterday's class, we already discussed why HGP is called the mega project. HGP, that is the information which are written in the entire DNA segments of human being, is about 3 into 10 to the power 9, that is 3 billion nitrogen bases. Each nitrogen base is either marked by A, T or C or G. So what information is required? That is 3000 books with 1000 pages, and each page should have 1000 characters. Then it will become 3000000000 nitrogen. Such a large information you go. चलो अब लिख लेंगे, कोई दिक्कत नहीं है. You got enough of time. जिंदगी बहुत बड़ी है, उसमें ये लिखने में कौन सी दिक्कत है. लेकिन when you want to retrieve it, when you want to a particular person, you want to know that is what is the gene for silk in the silk moth. So he may approach to such lab and they may say कि भाई वो लाइब्रेरी में जाना, सिल्क मोथ के बहुत सारे जीनोम लिखे हुए बुक्स पढ़ी है वहाँ पे 27 such books are there. From the book number 5, पेज नंबर 75, लाइन नंबर 35 से लेकर के बुक नंबर 14, पेज नंबर 6 से लेकर के वहाँ तक इंफॉर्मेशन. It will be such a huge information which one cannot carry. इतनी सारी बुक्स लेकर के जाएगा वो जस्ट टू नो अबाउट द सिल्क वर्म, जबकि पूरे पूरे कारखाने खड़े हो जाते हैं छोटी छोटी बुक्स के अंदर. इफ आई हैड टू एस्टैब्लिश अ कार मेकिंग कंपनी हियर, मैं अगर मान लीजिए किसी दुनिया की सबसे अच्छी कार कंपनी से मैं समझौता करूँ, मैं कहूँ कि भाई आई वांट टू स्टार्ट ए कार बिजनेस हियर इन इंडिया. So if I go and talk to them, what they'll give me? They'll give me some number of books. साहब ये ले जाना, इसके अंदर बॉडी के स्ट्रक्चर का सारी इन्फॉर्मेशन, ये ले जाना प्लांट कैसे खड़ा होगा उसकी इन्फॉर्मेशन, ये बुक ले जाना इसमें उसकी मशीन्स के बारे में सब लिखा है, ये ले जाना उसमें इलेक्ट्रिकल इन्फॉर्मेशन है. 4, 5, 6 books. बट ईच बुक इज कंटेनिंग वन मैटर, दैट इज वन थिंग, दैट इज द बॉडी शॉप है तो उसके अंदर सारी बॉडी से रिलेटेड जो भी चीज़ें वो आएगी. मैकेनिकल्स है तो उसमें सारी चीज़ें हाउ इंजन आर देयर, गियर आर देयर एंड ऑल अदर थिंग्स आर देयर, वो आ जाएंगे. अगर मैं कहते हैं इलेक्ट्रिकल्स हैं तो वो हेडलाइट से लेकर हॉर्न से लेकर के टेल लाइट से लेकर इनर लाइट से लेकर आउटर लाइट सारी इन्फॉर्मेशन रहेगी. हाउ स्टार्टर विल वर्क, हाउ द बैटरी विल बी चार्ज एंड दिस एंड दैट. बट हमारी बॉडी विच इज मच स्मॉल इन कंपेयर टू दैट कार, yet the information is spread in such a huge number of books, 3000 books, 1000 pages in each book and 1000 character on it. तो उसको रिट्राइव कैसे करेंगे? This was a very big problem way back in year 1900 when the computers were also in their initial stages. You remember now, first the storage information was stored on the floppy disk. Then came the hard disk, and that time hard disks were only of 1 GB, 2 GB. Then came CD, compact disc. Then came DVD, digital video or versatile disc. Then came cards, and now you got such small micro SD card, which is having information up to 128 GB or rather more. So I am talking about the way back in 1900. 1900 when such a huge information was there which has to be copied, retrieved, and the information whenever required you should get at once instead of going to the library and seeing from book number 5 to book number 16, from page number this to page number that. That would have been a very big hectic problem. Just like dystrophin, such a huge gene. Okay, so the other things may be small gene also, 3000 character का है तो 3 पेज में ही हो जाएगा, तो बहुत बड़ी बात नहीं है. So the different genes are of different sizes. Accordingly we'll have to see that. तो उसके लिए हमको एक नए ब्रांच की नीड पड़ी, that branch of the biology which deals or which link the information of the knowledge of the computer with the knowledge of biological processes. This is called bioinformatics, or the storage of the various informations of the nitrogen base sequence in the DNA and retrieved as and when required. That is called bioinformatics. Bioinformatics word was initially given for that NGP project, but today it has got vast scope. Okay, so that's all, and I think my time is also क्या 1 मिनट बचा है ना, 1 मिनट में क्या पूछ सकते हैं. You can ask me any question related to this chapter, else now tomorrow I'll have your sort of doubt class, any revision, anything required, because I am not starting any new chapter because for few days you will have exams. You want to ask anything, or should I put the video off? Okay, then that's all for today. Thank you.